evening to my cutest little champions out there. Well, today I have with me two very important guests. One is Mr. Amitabh Bachchan and the other person, can you guess her? Well, she is none other than Queen Elizabeth. And now this question might be lingering in your head, that is, ma'am, why are they actually over here? And I would not give you the answer to this question right away. Let it create suspense, okay? Think about it, keep guessing, and if at all you know the reason, comment down in the comment box below. I'll just give you a clue. It has something to do with the topic we are dealing today, and that is disorders in organisms. Well, this is the last topic of the chapter, and I'm pretty sure if you have been there with me throughout the sessions, you will get 13 on 13 from this particular chapter and 4 to 5 marks in your KCET examination. So if you have not watched these lectures yet, go watch it, put it in 2x speed. It will be definitely helping you really well. And for those of my lovely champions who have been so good to comment down the answers to all the questions I'm giving you, kudos to you. We have one new person out there, Seema Afreen. Kudos to you because you gave all the answers, lots of love. And then we have Gayatri Naik also, who have been consistent now in giving the answers, and my dear Sagar also, who had been giving the answers since long. I appreciate your consistency, my dear children. I never see all of this thing very usually. So it's very unusual to see your consistency, and let me tell you, keep going, okay? With this, will in your head, you will definitely achieve what others are not gonna achieve. Okay, you are a step beyond, a step beyond in comparison to all the other children who are preparing without any aim. So you guys have an aim, and I see that with your consistency. That does not mean others do not have, okay, but they do not realize that they had the potential. But now you guys know your potential and give your 100% best, okay? And I have Prajwal King DC and also Sagar who had that affirmation, and they put it down in the comment section also. I am going to rock PU2 examination. I asked who all has the affirmation. So yes, they have the affirmation, and I just wish that you give your best. And with that, we start off with today's lecture, and the first topic that we have is mutation. What do you mean by mutation? My dear children, is it good or bad? So some people already know that mutation is something that causes defects in the gene, or basically that alters the DNA sequences. My dear champions, do you know that this is our chromosome? The chromosome is made up of DNA, and the segment of the DNA is known as a gene. The gene, they code for what? They code for protein. Now suppose this segment of the DNA is altered. Okay, it's altered somehow. The structure is somehow altered okay, I won't complicate this. Somehow the structure is altered, which is why what will happen? Do you think this gene will be formed properly? No. If this gene is not there, do you think this protein is going to be formed properly? No. So basically this process where the alteration of DNA sequence is going to change what? The genotype, that is a genetic makeup, and also the phenotype, that is a physical character, is known as mutation. What is the phenotype over here? Whatever this protein does, my dear children, whatever is the function of this protein, that is getting altered, which is why I am telling you the phenotype is getting, disturbed. It is getting changed. It is not going to have the same effect that it used to have. So this process of alteration of DNA sequence is known as what? Mutation. Now there is a term given in NCERT which is known as point mutation. What do you mean by that? So I hope you know that the DNA which you see, my dear children, they are made up of certain nucleotide bases or nitrogenous bases out there. Okay, so nitrogenous bases as in what? As in adenine, as in guanine, cytosine and thymine. And I'll tell you one more thing. 3 of them come together to code for an amino acid. 3 of them come together to code for an amino acid. Similarly, the next 3 would be coding for what? Another amino acid. And when these amino acids, they join together, what is going to be formed? They are going to form a protein, which is why I said that DNA has the gene which codes for protein. So DNA are made up of segments like this, and each segment is going to code for some protein out there. Now when I say point mutation is happening, by that I mean that probably at some point over here, suppose a single base pair is getting changed. It is getting changed. From G it has become C. From G, it has become C. Then obviously, will it code for the same amino acid? No. If it is not the same amino acid, do you think the same protein is going to be formed? The answer would be no. So, whenever this type of mutation occurs, you call this as point mutation. And you are going to study about it really soon when I talk about sickle cell anemia. Okay, I'll teach you that. Now the next thing that we have is frame shift mutation. What do you mean by frame shift mutation? So over here I told you they are usually read in the number of 3. So this is 3, this is next 3. But suppose over here this particular nucleotide or basically nitrogen base is not there, it has been lost. So now will you read 1, 2, 3 together or not? You will be reading the next one, right? The next 3. So over here, where is it? So over here, instead of reading this particular thing, everything went up. Okay. So instead of reading this, okay, and coding for that amino acid, now this body will be reading what? A, C and T. Because G has been lost, because a particular nitrogen base has been lost, which is why what is happening, the frame of reading has been shifted, and whenever you shift a frame, what will happen, some other amino acid is going to be formed. Again, do I get the same protein if the amino acid is changed? Obviously no. So that is what I mean by frame shift mutation. It can happen because of 2 reasons. Either because of deletion, as I showed you right now, G is being deleted. Suppose apart from that, I can do one more thing. Suppose rather than removing G, I can add T over here. I can add an extra nitrogen base over here. In that case also the reading frame is going to change, because instead of reading AGC, now it will read as AGT. Because of what? One insertion of one nitrogenous base out there. So this is again an example of frame shift mutation. Remember, there are so many different types of mutations all together, my dear children. So these are the 2 examples given in your NCERT. Just have that in your head. And now the question comes, is mutation good or bad? Mostly it is bad, but sometimes it can be good as well. Suppose we have a gene that codes for a protein which is not actually great for our body. So that is getting mutated, okay, that is getting mutated. In that case, it probably might produce a protein which is not that bad. But again, that's a chance, okay, a chance that it can be good, but there's always a chance that it can be bad as well. So in our body, do you think mutation happens, or it is only happening in the people who have certain diseases? In our body, when I'm talking to you right now, so many mutations might be happening. But they might not have great effects out there. Why? Because my body is sensing that and repairing that. There is a proper DNA repair mechanism that is happening in my body. And when that also fails at times, everyone does mistakes, right? Similarly, in our body, the system also does some mistakes, which is why we do not realize that this is happening. Our body does not realize that this has happened, which is why we get certain diseases out there. And one very important example of this is what? Cancer. One very important example of this is cancer. Certain genes are getting altered over here, which is why our cells, which were normally dividing, now starts dividing very fast. They have some altogether very different characteristics which was not initially there. So cancer is obviously a result of what? Mutation that is happening within our body, and it can happen to anyone out there. But how do we keep ourselves safe from all these things? What is in our control? Ma'am, we should be keeping our mobile phone away. Why? Because these mobile phones which are there, they are emitting certain radiations. And what does this radiation do? They bring about changes like this. Okay, they bring about changes like this. They cause mutations, which is why what will happen? The genotype and the phenotype will change. Obviously, at the end, I might get some diseases out there. And so what do you understand from this? That phone is releasing some radiation which is causing what? Mutation. Now, since something is causing mutation, you have a name for it. These radiations which are released from the phone is what? It is known as mutagen. The one that causes mutation is known as mutagen. So UV radiation is sorry, UV radiation is also a mutagen, okay, the one that you get from your, what do you call, the sun and everything. We say that people who do not apply sunscreen can have a chance of skin cancer. Skin cancer rates are increased when people do not apply sunscreen. Why? Because of all these things. Okay, so if at all I said that phone gives UV radiation, I'm so sorry. Okay, there are so multiple types of radiations. I do not want to mess that up, but in general, just understand that anything that causes mutations out there, you call those causative agents as what? Mutagens. You call them as mutagens, and UV radiation is an example of that. Okay, and one such example, where do we get UV radiation from? From the sun, from the harmful rays of the sun. You get it? Which is why you need to apply sunscreen, my dear children. And now, going ahead with the next thing, all about the pedigree analysis. So, you might have heard about this term pedigree analysis, but what exactly does this mean? So, analysis of traits in a several of generation of a family is called pedigree analysis. That means, I am studying about some trait. I am studying about some character in a family. Probably it can be some disease as well. Okay, so when this analysis of traits is being done generation over generation, I call this as what? Pedigree analysis. Now, you might have seen this particular box given in your NCERT, which is very super important. Why? Because you have to remember all this thing, because you may get question as in they will be asking you, what do you mean by this? What do you mean by this? What do you mean by this? So, you should know the meaning. So, whenever it's a box like this, it means it is what? Male. Whenever it's a female, it would be circle. It would be what? Circle. Now, whenever you do not know the sex of the organism, you make it like this. Okay, it will be like this. Now, if you want to show that the male is affected, you color it. If you want to show the female is affected, you color this. If you want to show someone, I do not know the sex, but that person is affected, you show this case. So, whenever you color something, that means it is what? Affected individual. Now, there is male and there is female. When you draw a line between them, that means what? Mating is happening. What is happening? Mating is happening. Now, if I draw double lines in between them, double lines, that means they are very close to each other. What do you understand from this? That is, probably mating might be happening in between relatives. You know, you don't need to feel gross about it. The thing is that initially, okay, before so many years, it was like between the family members only people used to get married. Okay, uncle's daughter, uncle's son, and this and that used to happen a lot. And what do you call this? This is known as consanguineous mating. What is it known as? It is known as consanguineous mating. It's very important. This question is usually being asked. So, double line indicates that closer relation is there. You can what do you call, remember like that. This is just for your understanding, I am telling you. It's when mating is happening between the relatives. And when was it stopped? When was it stopped, my dear children? Because certain people, okay, in Kerala and all, in Attappady region, this is a region where so many tribal communities also live. There it was seen that so many children were having sickle cell anemia, a genetic disorder. So, then it was traced back that probably they were getting married between themselves, that is, the family members were getting married with each other, which is why this was, what do you call, the rate of it was increasing. So, something was there in the family and it was getting exchanged every now and then. You get it? So, which is why slowly when people got to understand the problems of this type of mating, my dear children, they stopped marrying within the family itself, which is why we generally say that you should not be marrying within the family, you should be marrying someone from outside. That is the actual reason. I hope you get it now. Okay. Now, over here, what does this particular mean? This particular thing means. So, there's a what do you call, square, and then there's a circle, there is a mating, and then out of this you get a square, you get a circle, and you get a square again. What do you mean by that? That means a boy and a girl is mating, and there are 3 children. One is boy and the other one is girl, and the other one is also boy. That is what you mean by this. Okay. Now, suppose I am coloring like this. Then means that means these 2 parents mated, and then there are 2 boys and one girl child. After the 2 boys, one guy is affected with some disease out there. Okay, one guy is affected with some disease. Since this particular thing is being colored, that is what it means. Now, what do you mean if it comes like this? If it comes like this, what do you mean? It means that 5 of the children are being produced and 5 of them are unaffected. I do not know the children, if the children are girls or boys. There are 5 of them and 5 of them are unaffected. Now if I draw like this, it means that 5 of them with unspecified sex, they are all affected with some disease out there. So whenever you color something, that means they have the disease. Okay, so in your NCERT they have given examples of certain pedigree chart, but I don't want to go ahead with that as of now. Okay, I will be telling you once I finish the diseases, so that it becomes more easy for you to understand it. Now, one homework question for all of you guys who have been listening to me: identify the correct choice for the given symbols. Okay, so there is A and there is B. What do you mean by A and what do you mean by B? The options are given over here. If you know the answer, put it down in the comment section right now. This is homework number 1. Cool? And with that, I think that's all over. Now we are going to go ahead with the major topic out there, which is disorders, my dear children. So we saw disorders are caused by what? Mutations. What are mutations? Phenomena that causes alteration in the DNA sequence. That's all. Okay, and we saw what is pedigree analysis. Pedigree analysis is the analysis of the traits being passed off generation after generation, and we saw certain figures out there. Now we are going to talk about disorders, where I am going to tell you there are 2 types of disorders. One is Mendelian disorders and the other one is chromosomal disorders. What do you mean by Mendelian disorders? So whenever I say that a gene is being altered, whenever I say that the chromosome and particular gene is being altered, and because of this gene or the DNA sequence being altered, I am not getting the protein that I wanted, and when this causes a disease, I will categorize it under what? Mendelian disorder. Okay, when it's just because of the genes, because it got mutated or just something happened over here, which is why now the same amount of protein or the same type of protein is not being produced, which is why I have a disease now. So I will categorize it under Mendelian disorder. On the other hand, suppose you need to understand one very important thing as of now itself, and that is chromosomes, before I go ahead with this. So in our body, how many chromosomes are there? In our body we have 23 pair of chromosomes. Okay, 23 pair of chromosome. What do you understand from that? That means, ma'am, I get one from my papa, another from my amma. So now I have what? 1 pair. 1 pair. So just like this, how many pairs are there? 23 pairs are there in my body. Each of the chromosome, 1 chromosome I'll be getting from my father, another chromosome I'll be getting from my mother, because both the gametes contain half the chromosomes. So half and half comes over here and becomes full when a zygote is being formed. So please understand, out of this 23 pair of chromosome, 1 pair of chromosomes are sex chromosomes. What are they? They are sex chromosome. And which one is that? The 23rd, the last one is the sex chromosome. So if at all, suppose, okay, I am getting 1 X chromosome from a parent and 1 Y chromosome from my father. In that particular condition, what happens? A child is born with XY chromosome. This is the last pair of chromosome that is there in the child's body. And this indicates that the child will be what? Male, because it was what? XY condition. Suppose it was X and X, then it would have been a female. So the last chromosome, the 23rd pair of chromosome, always remember they are known as sex chromosomes. Okay, the 23rd pair is known as sex chromosome, and you have a specific name for it. They are also known as allosome. They are also known as allosome. Ma'am, all the rest 22 of them, what do you call it? They are known as autosomes. They are known as autosomes. Ma'am, what do they do? So all these chromosomes have certain genes which specify other characteristics of my body. For example, my hair color, my eye color, my skin color, all the things, the amount of hemoglobin protein produced in my body, all the things are being, set aside by all these autosomes of my body, of my cell out there. So out of 23 pair, please understand 22 of them are autosomes, which codes for all the regular function of my body, and 23rd of them are what? Sex chromosome, which is responsible for my sexual characteristics. And it's not just sexual characteristics, they also have genes for other things also, other general things also. It's not like they code just for the sexual characteristics. They code for other things also, but majorly they are therefore coding for the sexual characteristics of my body. I know I am making it a whole fuss, but let me tell you, let me break it down, let me tell you once more, listen to me carefully. So after 23 pair, 22 of them, they code for general characteristics. They cannot code for our sexual characteristics. 23rd one, which we have in our body, is responsible for sexual characteristics, also some general characteristics of our body. I hope that's clear now. Okay, so the 23rd one is known as allosome, all the other ones are known as autosomes. Now, why did I give you all this information? The reason being, we have a different type of disorder which is known as chromosomal disorder, where what happens is that, suppose I said we get one chromosome from father, one chromosome from mother, but suppose we are getting two chromosomes from mother instead of getting one. So what happens, my dear children? Now I have three chromosomes out here. Instead of making a pair, I have one extra chromosome out here. This extra chromosome will have some genes also. They will also code for some proteins out there. So in my body, don't you think that there is an imbalance being caused because of some extra genetic material being added up? So much amount of protein is being formed, and that could also be lethal for us. Yes. So whenever this happens, whenever we have an addition of a chromosome, or probably from father we needed to get one chromosome, from mother we also needed to get one chromosome. That is how it works, right? But from the mother we got no chromosome at all. In the gamete there was no chromosome pair for the 22nd chromosome. In that condition, what do you do? Again, some protein is not being formed enough because there is only one chromosome to produce that. You get it? So there are disorders which is caused because of the chromosomal, what you call, structure, and also the numbers. So here I gave you the example of numbers. Now it can be structural also, where suppose this part of the chromosome is lost somehow. So whatever gene was there, that is also gone. You get it? So that can also be coming under chromosomal disorders. We are going to learn about each of them properly. As of now, we are going to start with Mendelian disorders first, where I'll be talking about chromosomes, and in the chromosomes I'll be saying that certain genes are not going to work and what is going to happen when certain genes are not gonna work. I am going to give you examples of certain diseases which comes under Mendelian disorders category. I hope that is clear to some level. At least some level. Okay, this topic is little complex, which is why you feel like all this is, but trust me, it gets easy with time. So this is the common difference that you have, and with that we move ahead with the first disease out there, that is color blindness. So color blind people can't see any colors. Do you think so? Do you think so, that they can't see any colors? It's not like that. It depends upon the, what do you call, intensity of it, okay, how badly they are affected. So basically what happens is that we have green and red cone cells which are responsible for helping us in distinguishing between the red color and the green color. So mostly whoever has color blindness, these genes which codes for these things are getting affected, which is why they won't be able to differentiate. Now there is another color blindness also, where they just see black and grey, black and white, there is nothing else in between, you get it? So there is different things, but this is something that is very common in people. Okay, so here in color blindness, or by color blindness I mean to say that there is a defect in the red or the green cone cells that is there in our eyes, okay, which is why they are not able to distinguish properly between red and green color which is there. So they won't be able to see the signals in your traffic signals and things like that. So now we are going to decode how does this particular thing happen. So over here I have written one thing, sex-linked recessive disorder. What do you mean by that? Ma'am, with this only I got confused. So I understand one very important thing with this is that these things, okay, the gene whichever is getting mutated, whatever has gotten mutated over here is on the sex chromosome, which is why it is sex linked. So I am making the mother over here and the father over here. If you listen to me carefully in this part, you will understand the whole thing about at least two diseases. One is color blindness and the other one is hemophilia, because both of them are equally same. So over here I have XX in the mother, 2X chromosome. The father I have XY. Okay, suppose in this X chromosome the gene that is coding for all this thing is getting affected. Okay, so it has mutation, so I am going to write it like this, XC. Fine. So this chromosome is affected because the gene part is somehow mutated in them. Now, whenever gametes are formed, either this X chromosome can go or Y chromosome can go in the gamete. Only half of them will be going. You know, half theory I just told you right now. Similarly, from the mother's side, either XC or it can be X, normal X that can be going. Suppose this and this comes together, the condition is like this. This and this comes together, condition is like this. Okay, suppose this and this comes together, then the condition is like this. This and this comes together, the condition would be like this. This and this comes together, the condition is like this. Now, what is the sex of this child? This will be a female. This will be what? A female. Does this female have color blindness? Does this female have color blindness? Anyway, she has the X chromosome which is mutated, right? Does she have it? Does she have the disease that is color blindness? The answer is no. Because, my dear children, in the case of females, if only one X chromosome is affected, there is another X chromosome to overshadow the effect, which is why they will still do the function and they will be just the carrier of the disease. So, this female was just a carrier. Similarly, the daughter which is being formed over here, she is going to be a carrier. Now, the next daughter, she did not have any problem at all. She did not have the mutated chromosome. She is just a normal person who is not even the carrier. Now, what about this guy? This guy also do not have anything. It's normal. What about this guy? What about this guy? There is XC chromosome which is affected and there is Y chromosome. Is there another X chromosome to overshadow the effect? No, it is not there. So, in this particular condition, don't you think the boy will be colorblind? The answer is yes. Since there is no one to overshadow the effect, the boy will be colorblind. And with this, you can link the other part of the story. This color blindness is sex-linked recessive disorder. Why is it sex-linked? You got to understand, because it is linked to some sex chromosome. Okay, the genes of the sex chromosome. Now, why is it a recessive disorder? Because, my dear children, it can only express itself in recessive condition. Suppose if the father also had a defect over here, he was also colorblind. Now, when this and this chromosome come together, it is this condition. This is where there is no proper another normal X chromosome. It won't be, there is no one to overshadow the effect. This is where the female will be colorblind. This is where the female is going to be colorblind. So, only in recessive condition it is going to express itself. Over here also it's recessive. Why? Because there is no other X chromosome. There is no other X chromosome, which is why we say this is also recessive condition. Okay, so recessive means when both the chromosomes are same, when both the things are same. But here, since there is no one to basically overshadow the effect, there is only one X chromosome and that is affected, which is why it shows the disease. Similarly, over here, if the condition was like this, then only the female will be colorblind, because this disease that we are discussing is what? A recessive disorder, and it is also sex linked. You will get to understand better. We will see some examples. So, over here, I have a normal father and I have a mother who is what? A carrier. Her one chromosome, X chromosome, is affected, the one that I showed you right now. So, I got a son. Okay, I got a son when I crossed this and this. So, So, I got a son when I crossed this, and that is this one, XY. So, he is normal. When I crossed this and this, I got this condition where the son was colorblind, my dear children. Where the son was colorblind. So, out of 2 children, 50% of the son, out of 2 of them, half of the boys, there are chances that half of the boys would be having what? Color blindness. So, this is given in your NCERT as well. A carrier mother has a 50% chance of passing it to her son. My dear children. So, this is the 50% chance. So, either she can form this gamete or this gamete. So, there is 50% chance for her to what? Pass it to the son out there. I hope you get it. Now, if you talk about the daughters, okay, here one is a carrier, another one is normal, just like how I showed you. Okay, I showed you here itself. I showed you by cross. I'll show you another example, okay, another condition altogether, where what is happening? The father is normal and the mother, my dear children, both her chromosomes are getting affected. That means she is having what? Color blindness. This is recessive condition, no? Where both the chromosomes are same, both having the same effect, right? So this is what? Color. She is having color blindness. Now the gametes are X and Y. Here the gametes are XC and XC. In this condition, what will happen, my dear children? Anyhow, whatever you do, the boy will be having color blindness, because both the X chromosomes are having the what do you call the mutated gene out there. So whatever you do, one X chromosome have to come over here to make a boy. To make a boy, you need one X chromosome from the mother. So both the X chromosomes are gone. So in that case, my dear children, anyhow, whatever you do, the boy will be affected. Let's see it. So suppose I fuse this with this, I get this condition. That means she is a carrier female. Okay, only one is there, so she is a carrier. Now this and this, again, she is what? A carrier female. Suppose this goes with this. He is what? He is a colorblind male. Suppose this goes with this, again he is what? A colorblind male. Perfectly. So all the boys who are going to be produced, 100% of them are going to be what? Colorblind. Are going to be colorblind. If we talk about the females, 100% of them are going to be carriers in this particular scenario. So there are multiple scenarios and you need to know that. Okay, so you should know when is a female going to be colorblind. You do not say she is colorblind until she has 2 chromosomes like this. Otherwise, you always say that she is a carrier. Okay, so for a guy to be colorblind, okay, you just need the mother to be a carrier. There are 50% of chance that yes, the guy can be colorblind. Among the boys, if we see, there is 50% of chance. If we see in a whole, there is only 25% chance. 25, 25, 25, 25. 25% chance. But if we talk about just the sons, then out of them, 2 of them, one is getting affected, so 50% chance. Okay. Now another scenario altogether. You have colorblind father and the mother is normal. In this condition, what will happen? Since the mother is normal, here she will be contributing X, and from father's end you get Y, okay, for a child to be born as boy, okay. So since here it is normal and the Y is obviously going to be normal, my dear children, the sons are going to be pretty normal. What about the daughters? Over here, since only one X chromosome is getting affected over here, and on this side there is no X chromosome getting affected, whatever combination you make, my dear children, it will be always what? A carrier combination only. They will only produce carriers, nothing else. And the boys are also going to be safe. So that is what is being mentioned over here. Okay, that is what is mentioned over here. So females are going to be carriers in this condition, but the males are going to be completely safe. Now there is one more scenario where the mother is colorblind and the father is also colorblind. In this condition, what will happen? In this condition, what do you think will happen? So anyway, whatever, you don't need to cross it. Now I hope you know that. Okay, so this and this Y comes together. One X chromosome is affected anyway, the guy would be colorblind. This and this comes together, again it will be colorblind. Females also, since father is contributing X chromosome and that is affected, and the mother also has 2 affected X chromosome, whatever you do, the child, all of the children will be what? Colorblind itself. All of the children would be colorblind. Okay, but if you have seen it, my dear children, if you checked it over here, so here, okay, whatever you do, the females are just going to be carriers mostly. Here the males are not affected, but the females are just carriers, they do not have this disease. Here the males are actually affected, but females are still the carriers, they are not affected. Here in this condition also, the female is a carrier, but the male is getting affected. With this we understand one very important thing, and that is what? That the chances of males to be colorblind is more, because this is what? X-linked recessive disorder, where it is about the X chromosome and you have a normal X chromosome. I mean, females have 2 X chromosome, and if they have one normal X chromosome, that overshadows everything. So it is mostly found in the case of males. Males are more prone to have this. So a very important line is given in NCERT. Please remember this very important line. That is, it occurs in about 8% of males and only 0.4% of females. 8% of males, the number is important. 8% of males and only 0.4% of what? Females. Females are usually carriers unless they inherit 2 defective copies, one from each parent. Okay, they are usually carriers unless and until they get 2 copies, one from each parent, defective copies, one from each parent. Probability. A carrier mother has a 50% chance of passing it to her son. I hope you got it. Why? Because carrier mother has one normal X chromosome, another defective X chromosome. So 50% chance are there that it can go to each gametes. A daughter will be colorblind only if her father is affected and the mother is a carrier. Okay. A daughter can be colorblind only when the father is definitely having it, one X chromosome, defective X chromosome came from him only, and the mother at least have to be a carrier. The mother has color blindness, then also the daughter can be colorblind, but even if she is a carrier also, then also there are chances. We saw that over here. We saw that over here. I'll show you. If at all you're forgetting things, over here see the father is colorblind. Okay, the mother, okay, wait, not this one. Where is it? Come on, where did I? Is that condition not there? Wait for a second, okay? Okay, that condition has not been discussed. I'll discuss with you. So over here, suppose this is the mother and this is the father. The father is colorblind and the mother is a carrier. In this condition, you will get at least this thing should be there for a female to be colorblind is what they are saying, because this and this will come together and there are chances that the female can be colorblind. Other way that the female will be colorblind is when the father is colorblind for sure and the mother is also colorblind for sure. Here there are 50% still chances, 50% chances for the daughter to be a carrier, but here there is no % chance. They will be defective anyhow, whatever you do. Okay, why 50% chance over here? Because this and this, when it comes together, you get a carrier female. Okay, not a defective female. So I hope that clears up everything. Okay, so what did we learn about color blindness? So color blindness, my dear children, is what disorder? Is sex-linked recessive disorder. It is when there is defect in the genes for red or green cone cells of our eyes, which is why it is difficult to distinguish between red and green color. It mostly affects the male because it's a recessive disorder. Okay, if it was a dominant disorder, what happens in dominant disorder? For example, if it is dominant disorder, even if one chromosome is affected, it will show away its character. But since it is not a dominant disorder over here, which is why they are all carriers. If it was a dominant disorder, probably the females would have been having the disease with the same genotype up here. Okay, so this is a recessive disorder. 8% of them are mostly males, 0.4% of them are mostly females who have the disease. And a carrier mother has 50% chance of passing it to the son. Remember that. And the daughter will be colorblind only when the father is colorblind and the mother is at least a carrier. Cool. And now comes the second sex-linked recessive disorder, which is hemophilia. And there's a question being put up, which is that, did Queen Victoria had hemophilia? Well, who is Queen Victoria? She is the great-great-grandmother of Queen Elizabeth, my dear children. And you know what? She was actually, Queen Victoria was actually a carrier of hemophilia. And since she was a carrier, there's so many people in the family had this particular thing. Okay, it got passed on, and people had it. One of her child, his name was King Leopold, I forgot. Okay, he also had it. He also had hemophilia and he passed away. So basically this was a thing and I found it very interesting. It was given in your NCERT, which is why I thought I should be talking about that as well. But what happens exactly in hemophilia, which is why is it known as very dangerous? Okay, is it actually dangerous? Let us know all about it as of now. So over here, my dear children, what happens is that a gene that codes for a protein which is involved in the clotting of the blood, okay, which is very much important for the clotting of the blood, is getting affected. So this gene is getting affected, which is why there is no protein, and since there is no protein which is available, clotting of the blood becomes a very difficult task, which is why even if a simple cut is there, my dear children, it will keep on bleeding on a non-stop basis. You get it? So that is the problem with hemophilia. Again, can I consider it as a Mendelian disorder? Yes, because what? A gene mutation is happening over here. Now, why is it known as sex-linked recessive disorder? Because again, it's on the X chromosome. It is there on the X chromosome. Now you tell me, if it is there on the X chromosome and it is a recessive disorder, there is chances for males or females to get this disorder? More. It will be obviously males. It would be obviously males. And you know what? Once, it's not like color blindness where you cannot differentiate color. It is actually very dangerous for the human, because suppose blood is just going. Obviously, can someone survive out there? No. It is not very easy to survive in that case, my dear children, right? So which is why people who generally have hemophilia, they don't survive until long. Now there are medications and things like that, modern treatments are available, which is why there are good things happening in the body, my dear children. But initially, I'm talking about long back, okay. So that particular point of time, a person, if they had hemophilia, I'm not talking about carriers, I'm talking about people who have hemophilia, they were not able to survive. And I'll give you one more reason why it is always males and why it is not females. Because, let me decode that. So first of all, I have to go with the basics first. So over here, the first condition is where the female is a carrier. Female is what? She is a carrier. Again, this and this comes together. The daughter is a carrier. This and this comes together. The daughter is normal. This and this comes together. You have an affected son. This and this comes together. This is a normal son out there. So that is okay. But over here, if you see, the son is getting affected. Okay, again a condition. Suppose the male is fine. The female, she has hemophilia. Female, she has hemophilia. In that condition again, suppose this and this comes together. Carrier. This and this comes together. Again a carrier. This and this comes together. Again a male who is having the disease because there is no X chromosome to overshadow it. This and this comes together. Again a male who is having the disease because there is no X chromosome to overshadow it. And whatever condition you do, my dear children, you will get that the males are the ones that is mostly affected. And why are the females not affected? So in this NCERT, there is a line being mentioned where, my dear children, it made sense also. So for a female to be affected, the male should also have this disease. Agree? Suppose the condition is like this. Females can be a carrier. So they will be there, they will be alive since they are just carriers, they don't have this disease. Suppose the condition is like this. But the males, they have hemophilia, and with the hemophilia, do you think they can live longer? Do you think they can live longer to reproduce? The answer is no. Which is why again the chances of female getting affected is very, very less. Because only in this condition, when the males are affected and when they actually mate with another female who at least is a carrier, then only this and this can come together to produce a female who is having the disease, but they are not alive. Only then what will you do? Obviously the chances becomes so less. So in two conditions it can happen, the females can be affected. When either she is a carrier and he is basically having the disease, anyway he have to have the disease and she should also have the disease. But in this condition the female also won't survive. In this condition the female also won't survive. So female can survive only in this condition, that is where she is just a carrier. But then also the babies would not be formed. Why? Because the male partner would not be there who is hemophilic. They won't be alive itself. You get it? So that is the funda. I hope you understood it. Since it is not like color blindness where you can live with it. Hemophilia is something where you have the disorder, probably you are in danger of death as well. Now things have changed, I am telling you again and again. So possibility of female becoming a hemophilic is extremely rare. Very important question. Because mother of such a female has to be at least carrier and the father should be hemophilic. And I explained you right now why. Okay, because you need XX chromosome from both of them. So if X chromosome affected in male, my dear children, that male is hemophilic and he cannot survive. Unviable in the later stage of life. So that is written over here. And that is rare because why? The father will be unviable in the later stage of life. That means that person would be dying off. He won't be surviving through all the initial phases of the life. So that they can go for ahead with reproduction. I hope you got it. So understand one important thing. Queen Victoria didn't have hemophilia, she was just a carrier. She was just a carrier. Okay, now Queen Elizabeth, did she have it? The answer is no, she didn't have it. Okay, she didn't have hemophilia. Now, if we talk about Queen Victoria's son and things like that, as I told you, Leopold was there. At 50% of chance for a carrier to give rise to the son, what do you call, pass the disease to the son is there, right? We discussed in color blindness and this also. So the same thing happened. Leopold who was there, he got the disease and he died off. Okay, so that is about it, and that's all. So those are the two sex-linked recessive disorder, and now we are going ahead with the second type of recessive disorders. All of them, my dear children, please understand, they are all linked to Mendelian disorder. They come under the category of Mendelian disorders where the gene are getting mutated. But this is not sex-linked, this is autosome-linked. That means the genes that we are going to talk about now, they are not on the sex chromosome, they are on the autosomes out there, and they are also recessive. That means if only one gene is affected, then it's okay. If multiple genes are affected, my dear children, then that becomes a problem. So, people with sickle cell anemia won't have malaria? Well, I'll answer to that later. But first of all, we are going to talk about sickle cell anemia as of now. Why is it known as sickle cell anemia? First question. Because the RBC, my dear children, which is there, it is having a shape like, it is known as bioconcave shape. Okay, what is it known as? It is known as biconcave shape out there. It's a discoid, what do I call it? It's like this. It's almost like this where you have a bend in between and things like that. But this RBC carries hemoglobin, right? And when the hemoglobin is not formed properly, as in there is some problem in the structure of the hemoglobin. Hemoglobin is a protein, and when there is a problem in the structure of the protein, what will happen? Basically, the oxygen carrying capacity of hemoglobin is reduced. So, in general, let me tell you. So, we have RBC. RBC carries hemoglobin, and hemoglobin is responsible for carrying oxygen. If the structure of hemoglobin is not that great, do you get oxygen? Do you get enough amount of oxygen out there? No. And under this low oxygen tension, my dear children, what will happen? Under this low oxygen tension, the shape of the RBC, it gets changed to what? Sickle shape. It gets changed to sickle shape. You get it? So, what might be probably getting affected? The genes that are coding for what? Hemoglobin. The genes that codes for hemoglobin is getting affected. Hemoglobin is a protein, I told you, and that is getting affected over here. Where are these genes? On the autosomes. And what is exactly happening with the gene is also being mentioned over here for the first time. So, you have to listen to me carefully. So, please understand, when you talk about hemoglobin, it is made up of two things, alpha globin chain and beta globin chain. So, two things makes up hemoglobin. One is alpha globin chain and the other one is beta globin chain. So, for alpha globin protein, there is another set of genes. For beta globin proteins, there is another set of genes. Both these proteins comes together to form hemoglobin out there. Okay, or both these chains, not proteins, they call them polypeptide, the shorter form of proteins. So, both these polypeptides come together to give rise to hemoglobin, which is a big protein out there. For easy understanding, I am telling all this thing. Okay, so what exactly happens is that there is a mutation in the beta globin chain. In the beta globin chain out there, there is a mutation. What exactly happens? The thing that happens is that, suppose you have nitrogenous bases. I told you initially, right? A, G, C, T and things like that. So, you have nitrogenous bases. One over here, three of the nitrogenous bases will be over here. So, this is one group, and then another group, and then another group, third one, and then another group, and then another group, and now you have the sixth one. In the sixth one, my dear children, what happens is that usually over here, it should be CTC. It should be what? CTC in the DNA. And then from the DNA, something else is being formed first, and then only a protein is being formed. What is this something else? It is known as an mRNA. You will study about this in the next chapter. Okay, something else is being formed, and that is mRNA. So, over here, the DNA which is having CTC, okay, it is getting transcribed to an mRNA, where C will be going with G, T will be going with A, and C again will be going with G. So, in the mRNA, we should be having GAG. Understand this fact that A pairs with T, okay, A pairs with T, G pairs with C. This is the usual trend that we see. A goes with T and G goes with C. This is in the case of DNA. DNA is double stranded, so probably here if it's A, then here it will be T. That is how it works. Now, if the DNA is forming an RNA over here, then in RNA you do not have T, you have U. You will be having U. So over here, C is going to pair with G only. T, as I told, T is there in the DNA, it will be pairing with A over here, and C, which is again there, it will be forming G over here. So this will be coding for some amino acid over there. But now there is a point mutation happening. Point mutation, remember, just only one single base pair is getting changed over here. So instead of having T over here, suppose there is, did I do that? Okay, instead of having T over here, instead of having CTC, it is now CAC in the DNA. In the DNA it is what? CAC. Then my dear children, in the RNA, when it forms, CAC forms the RNA. How does it form? So C goes with G. What does A go with? I told you A can pair with T in the case of DNA, but in the case of RNA it pairs with U. In the case of RNA it pairs with U. So A will be going with U over here. Again, G pairs with what? G pairs with C. So C will be going with G over here. So it becomes GUG. So instead of having GAG, we are having GUG over here. That means a whole, a codon is being changed over here. This particular thing is going to code for some another amino acid. Over here, this was coding for some another amino acid. What is this amino acid, my dear children? So it was initially coding for an amino acid which is known as glutamic acid. It is known as glutamic acid. But now since this change has occurred, my dear children, it is now going to code for valine. It is now going to code for valine. This is the change that happens. And now since one amino acid change, the whole protein is going to get changed, which is why the beta globin gene which is there, it is not going to produce the exact beta globin which it was producing initially. There is some change, which is why there will be change in the structure of hemoglobin also. If there is a change in the structure of hemoglobin, low oxygen will be there. If low oxygen is there, the shape of the cell will turn into what? Sickle shape. You get it? It will change into what? Sickle shape. I know it's kind of confusing. Okay, and I can probably think of that by myself as well, because I was also a student once. So I'll explain it to you once more. Okay, this time no hodgepodge. So listen to me, one very important thing. Okay, starting from the basics. I'm so sorry for wasting away time, but yes. So this is a chromosome, right? This chromosome has the DNA and the DNA has the genes. So over here, there is a DNA strand like this. So usually it is like this, okay? Where suppose it is A over here, then A nitrogen base pair usually goes with T. So over here on the second strand, it will be T over here. And G usually goes with C. So suppose here it is G, then it will be C over here. Suppose here it is C, then it will be G over here. It goes like that. In that way, you have 1, you have 2, you have 3, and now you have reached 6th position. Which position? 6th position. And now here it was CTC initially. It was what? CTC initially in the DNA strand. I'm talking just about one DNA strand over here as of now, because only this one DNA strand undergoes a process which is known as transcription, about which you're going to soon study in the next chapter. Undergoes a process which is known as transcription, where out of this DNA I am going to make an RNA which is known as messenger RNA. The problem with RNA is that RNA does not have thymine in it. There is no T in it. RNA will be having U in it, that is uracil nitrogenous base. Okay, so whenever it sees T, it will be giving A, that's not a problem. But if A is there, it will be giving U. So I will be showing you example also. So over here it is CTC. So C goes with G. So whenever you are making an RNA out of this, so C will be going with G, T will be going with what? A, my dear children, and C again will go with G only. So this GAG which is there, which I form in the RNA, it is going to code for an amino acid which is known as glutamic acid. Now, second scenario is when, this is the first scenario, right? Now the second scenario is when in the DNA, it is not CTC, it is CAC. It is what? CAC. There is a mutation happening over here. Single point mutation is happening over here. Now it is CAC. In this condition, in the RNA, what is going to be formed? So C goes with G. A goes with what? A goes with not T but U, my dear children. Okay, and C again goes with G. Now here, instead of forming GAG, I am forming GUG, which codes for what? This codes for valine, which is another amino acid. Now since there is change in one amino acid, my dear children, the whole protein structure becomes changed, which is why the beta globin change that I am going to get, it's not the normal one. It's not the normal one. And since this is not the normal one, it will pair up with the alpha globin chain and form a hemoglobin with a destructed structure, which is why it is not able to carry enough amount of oxygen, and which is why the cell's shape become what? Sickle shaped. I hope you got it. So in your NCERT, they have not mentioned about the DNA and things like that. They have directly mentioned about GAG and GUG. They have said that GAG, there is a point mutation over here. A, instead of A, it is U. So which is why instead of glutamic acid, valine is being produced. Well, I'll tell you easy way to remember it. Okay, you don't have to remember CTC and CAC. It's for your understanding that I told you. Okay, but easy way to remember GAG and GUG is that in your alphabetical order A, B, C, D, A comes first and U comes later. So it was A initially, but it was replaced by U. Now in your alphabetical order, G comes first and then comes V. So initially it was coding for glutamic acid. Now it is coding for valine. Where is this happening? On the 6th position of the chromosome. Okay, on the 6th position of the gene, this particular thing is happening. If at all you want to take a screenshot, you can take it. Okay, this is the RNA, mRNA. Cool. Now, where are we? We are over here. So, we studied out that how the genes and everything is getting affected. Now we need to know what are the chromosomes that we need for this particular disease to occur. Okay. So, suppose from the mother's side, I am getting a gene HBS. Okay, or probably I'll make it easy for you. So, over here I had normal gene. The normal gene is represented by HBA. So from the chromosome of the father, I get HBA gene, and from the mother also I get HBA gene. Then it's totally fine. Then it's totally fine. I will be normal because I have two genes which are normal. But this is known as homozygous condition. This is what? Homozygous condition, where both the genes are same. Now, suppose I have this condition where from the father I am getting a defective one, sickle cell anemia one, and from the mother I am getting a normal one. Okay, in this condition, I have HBS and HBA, one normal and one that is affected. Am I a carrier or do I have the disease? The answer will be, I would be just a carrier. Why? Because it's a recessive disorder. Both of them should be there. Both of them should be defected. Then only I will have the disease. Otherwise, I will not have it. So, in heterozygous condition, I am not going to have the disease. I will be just the carrier of the disease. Okay. Now, the third condition which is there. The third condition is where, my dear children, over here, where should I write? Okay. HBS from father, HBS from mother. Both the genes are affected now. Both the genes from mother and father are affected. In this condition, I would be having the disease that is sickle cell anemia. That is what? Sickle cell anemia. So, HBS and HBS homozygous condition, there will be diseased individual. HBA and HBS heterozygous condition, I will be just a carrier, unaffected, but I can pass the gene to the next generation. HBA and HBA normal beta globin chain will be produced. If, one more thing is written, if both the parents are carrier, the child will also have it. Suppose, what do you mean by both the parents are carrier? Suppose one parent is like this, having, all of us has 2 chromosomes for each thing, right? So I have 2 chromosomes. Again over here HBA and HBS. This and this comes together, HBA, HBA, normal child. This and this comes together, HBA, HBS, carrier child. No female, no male, it can be carrier, that's all. Listen, this is not sex linked, okay? This is autosomal, which is why there is no sex being mentioned over here. It can be anyone, okay? Now, this and this, HBA and HBS, again what? A carrier. this and this, HBS and HBS. That means what? This person would be diseased. So if both the parents are carrier of sickle cell anemia, the child can have the disease, which is what I was talking about in Kerala, in a region known as Attapadi, and not just in Kerala, so many area it happened, okay? In Attapadi, lots of people were having sickle cell anemia because of the same reason. In the tribal community, people were getting married, they didn't know that they had the disease, they were just the carriers, so how will they know that? But their babies were dying. Why? Probably because of this particular thing, that the child could have got sickle cell anemia. They were not just the carrier, they probably might have got sickle cell anemia. I hope you got it. Okay, so please remember all these crucial details. How do you remember it? You cannot remember line by line of NCERT. You can only remember if you remember all this thing, HBA, HBS and things like that. You have to imagine. Then only you will be able to remember this. Now a question that I have put up is, people with sickle cell anemia won't have malaria? The answer is yes. Usually if I am affected with sickle cell anemia, I would not have malaria. Why? Because malarial parasite stays in the RBC. So they won't be able to stay alive. That is why people would not have malaria. Even for carriers also, if someone is carrier, usually they are resistant to malaria. Okay, some of them are not resistant, but usually talking, they are mostly resistant to malaria. Why? Because even carriers will be having some defective, what do you call, things out there, right? And in this defective condition, obviously it cannot survive. I hope you got it. Cool. So this is the thing about the first autosome linked recessive disorder. We studied two sex linked recessive disorder. One was hemophilia and then the other one was color blindness. And now the first autosome linked recessive disorder, which is sickle cell anemia, where the beta globin gene is getting mutated. Over here, GAG is getting converted to GUG. This is the GUG. This is the example of point mutation. Glutamic acid to valine, and the whole protein is getting changed. Okay, I hope you got it. And what else? Yes, and also one more thing. So here the quality of the beta globin chain is getting affected, right? Is it the quantity or the quality? The quality is getting affected. So always remember, this is a qualitative disorder. What is it? This particular disorder that you see is a, we will come to that again, but still, qualitative disorder. Beta globin chain's quality is getting disrupted, or hemoglobin's quality is getting disrupted. Cool? Now, should I teach you this now or I should go with this? I will go with this, that is thalassemia. And here comes the next thing. Amitabh Bachchan is a thalassemia carrier. What do I mean by that? What is thalassemia and why did I choose this after sickle cell anemia? Because both of them are blood disorders. Both of them are blood disorders. Okay. And there is a minute difference. So over here, in the case of sickle cell anemia, only beta globin chain was getting affected. Right, in thalassemia, my dear children, it can be either alpha or beta, or both of them getting affected. Okay, there can be alpha, beta getting affected. So here, the mutation or deletion reduces synthesis of either alpha or beta globin chain. Understand this thing. Over here it is written, it reduces the synthesis. It is not saying that structural abnormality is happening in alpha or beta. It is saying the alpha or beta chain production itself is reduced. So it's a what? A quantitative disorder. Enough amount of alpha beta globin chains are not there. Now it is a quantitative disorder, that means enough amount of hemoglobin is also not going to be formed, leading to abnormal hemoglobin and what? Anemia. Anemia is a condition where you do not have much amount of hemoglobin in the body, iron in the body. Okay. Now alpha thalassemia, what happens in that? Affects the alpha globin production caused by mutations in the HBA1 and HBA2 on the chromosome 16. So you have to understand this. This is chromosome 16, but do you have a pair? Yes, ma'am. So over here you have a gene. This is HBA1. You will be having it here also on the second pair, HBA1. And then there is one more gene, that is HBA2. This is HBA2. This is which pair of chromosome? This is chromosome number 16. This is where you have the genes which codes for the alpha globin gene. Now suppose this gene is getting affected. That means little reduction in the alpha globin will be there. Suppose this is also getting affected. Little more reduction would be there. This is also getting affected. More reduction would be there in the production. This is also getting affected. No alpha globin chain is being produced at all. You get it? So this is why I say it is a quantitative disorder rather than that which was a qualitative. There some mutation was happening which was changing the structure of the protein, but here the protein itself is not being formed. I hope you get it. Okay. And this gene's name you have to remember, HbA1, HbA2. So there is a pair, so obviously HbA1 will be here also, HbA2 here also, and also here. Do not go with HbA1 here, HbA2 there. It's not like that. Whatever gene you're talking about, it will be having 2 alleles on both the chromosomes. Okay, so this is one gene, HbA1, and this is HbA2, that is the second gene. Now, there is beta thalassemia. It affects the beta globin production caused by mutation in the HBB gene on the chromosome 11. So there is another chromosome, which is chromosome number 11, and over here you have just a single gene, that is HBB gene. Okay, here HbA was there, right? Here HBB is there. There is only a single gene. Suppose this is not there, that means little amount of beta globin production. If this is also not there, no production of beta globin gene. So alpha thalassemia is caused by when this all things happen. Beta thalassemia is caused when all this thing happen. So how do you remember chromosome 16 and 11? Always remember 16, it looks somewhere like B, right? Somewhere like B, but it is not for beta thalassemia. It is actually for alpha thalassemia. You can remember like that. Or probably since alpha it has an A, if you put like this, it becomes 6. So 16th chromosome is for alpha thalassemia, and the chromosome number 11 is for beta thalassemia. My dear children, I know it might be confusing you way too much because you studied a lot of things, but trust me, if you complete this up, it is going to help you really well. Okay, so this is about beta thalassemia. Now, over here I have a picture. So chromosome number 16, I told you it have HbA1 gene. If you are not able to see, I will show you. HbA1 gene here and here, HbA2 gene here and here. Okay, this is chromosome number 16. Now, when do you say that you are a carrier? Basically when there is one gene which is getting mutated, only one. Okay, alpha thalassemia minor. This is when basically 2 genes are getting mutated, but you still have 2 normal ones, which is why you are surviving with that. Okay, so you are minor case, you have mild thalassemia over here. And these people are just like, they are all carriers. They will be all like carriers. You are carrying the disease, but you are not showing it very much. This will still show it, but not way too much. It's mild thalassemia. Over here, when this and also this is getting affected, it will be what? Moderate to severe thalassemia, which is there. And when all of it is affected, you call this as thalassemia major. In this condition, a person would not be able to survive after a certain period of time. For example, 10 years. You get it? So that particular thing is there. Okay, so this is about thalassemia. And is thalassemia a disease that anyone can have? Can you be a minor patient of thalassemia? The answer is yes. Anyone can have it. So what do you need to do? Do the genetic checkup. Being done. So what happens, my dear children, is that now the actor, the celebrities are very much encouraging about awareness of thalassemia and every genetic disorders. Is that why they do that? Is that because people do not realize it, and then they have kid and then they lose the child because that child dies of the disease out there. So it's better to have the checkup already being done. So that is the thing. I'll tell you, in my family itself, okay, my uncle had thalassemia, and probably the auntie whom he got married to also had thalassemia. They were both carriers of thalassemia. Okay, probably both of them were minor cases of thalassemia. When both the minors came in, my dear children, what happened? Suppose this chromosome came from the uncle and another same type of chromosome came from the auntie also. The child, both their children had thalassemia. Both their children had thalassemia. Now that takes to the question that, ma'am, if the uncle had thalassemia, does your mother's family also have, or probably whoever he belongs to, does they also have thalassemia running? Probably yes. Which is why it is there. You get it? So that is something very, very important, and that is why you are studying about all of this thing over here in 12th grade. Otherwise, there is no point of studying all this thing, right? So I hope you got it, and with this, I hope you understood one more thing, that this is a quantitative disorder. It is talking about quantity, not quality. Okay, and the same goes with beta thalassemia. So over here only one gene for beta thalassemia, HBB. Okay, that is the one that codes for beta globin chain, and that is, if at all one is getting affected, carrier. If both of them, major case. If one is there, then it's minor case. If both of them, then this is a major case. So there are so many people who are thalassemia minors, and one person is over here itself. So if the other partner is okay, then will they have thalassemia? No. then the chances of your child to probably be a carrier would also be there. You have to check that. Okay, that's all. So, but yeah, regular checkup is very, very important. So that's about thalassemia, and I hope you got it, how it is different from sickle cell anemia. I hope you got it. So there's a different thing over here. I have mentioned it. Where is it? Yeah, which one is quantitative and which one is qualitative? So sickle cell anemia is qualitative because you're talking about quality of beta globin chain. Okay, the structure is getting altered, the quality is getting altered. Why have I written hemophilia here? Sorry, this is thalassemia. In thalassemia, the quantity is getting deteriorated. That much amount of quantity is not being produced. Hemophilia is also a blood disorder. Okay, but it is sex-linked blood disorder, which is why it is different from both of these guys. They are autosome-linked recessive disorders. They are also blood disorders, but that is autosome-linked. Now we have one more autosome-linked disorder, and that was none other than, what is it? Phenylketonuria, about which we already have studied. And here there is no much, problem in remembering this, hopefully, because we studied this in, pleiotropy, right? But anyway, I'll tell you. Okay, so what happens is that again the chromosome, the gene that codes for a very, very important protein, which is phenyl, I'll write it properly so that I don't have to write it again. That gene codes for an enzyme, a protein. Listen children, enzymes, are they protein? The answer is yes. Okay, so if I tell you that the gene codes for some enzymes, don't get it wrong, it's like gene is coding for some protein. So enzyme is also a protein. So this enzyme, phenylalanine hydroxylase, it is an enzyme that converts phenylalanine to tyrosine. So phenylalanine is an amino acid and tyrosine is also an amino acid. But what happens is that since this gene is not there that codes for phenylalanine hydroxylase, do you think this conversion can take place? The answer is no. And what happens to this guy? It is getting accumulated in the form of phenyl pyruvic acid. It gets accumulated in the form of phenyl pyruvic acid. And when this is there in too much amount in our body, what happens? Mental retardation takes place. Okay, so accumulation of phenylalanine or phenyl pyruvic acid leads to what? Mental retardation. And I'll tell you why this is also an example of pleiotropy. I'll just brush up that thing. Pleiotropy means one gene having multiple effect. So one gene that is coding for this has multiple effect. One effect is this. Other effect is that without tyrosine being formed, my dear children, melanin is not being produced. Melanin is not being produced, and when melanin is not there, hypopigmentation will be there. Okay, your hair will be grey in color, face will be too much fair. Okay, so that is hypopigmentation. Reduced skin pigmentation will be there when melanin is not there. Melanin is produced when tyrosine is there. So since this conversion is not happening, this character is also getting altered. So which is why it is an example of pleiotropy also. And it is an autosome linked recessive disorder, which means that the gene that codes for this is on autosomes, not sex chromosomes. Okay. Now the compounds which are there, for example phenyl pyruvic acid, they are actually excreted through urine also, because even our kidney cannot absorb it back. You get it? So there are so much bad things happening up there. So that is it. Yeah, that is phenylketonuria. And with that, we have covered all the Mendelian disorders. 2 of them were sex linked, 3 of them were autosome linked recessive disorders. All of the things that we studied until now are recessive disorders. In your NCERT, only one example is given about the dominant disorder, autosome linked dominant disorder, and that is what? Let's see it. I have put that over here. Okay, and that is myotonic dystrophy. So this is the pedigree chart. I told you I'll explain you the pedigree chart. So here it goes. Listen. So over here, I am saying it is autosomal dominant trait, that is myotonic dystrophy, right? So this is the female and this is the male. Similarly, this is the male and this is the female out there. Over here, difference, okay, which will help you very, very well is that in the parents you see that they are not diseased, but in the children they are diseased. In the parental generation you see they are not diseased, but in the children you see they became diseased. That means it is an example of what? Recessive trait. Because in recessive condition, my dear children, they can be just carriers, which is why they don't show the disease. They are recessive. Only if one chromosome is affected, it will just stay there like that. There is another chromosome to overshadow it. That's why it's recessive. So in the parental generation, it won't be seen. So over here, again, my dear children, see, these two are parents, but again, in their children, the disease is there. With this, we understand to spot recessive disorders. So when there is skipping of generations, you understand that you are talking about recessive disorders. Now I'll tell you, when do you say autosomal and when do you say sex linked. So when you see that more of the boys are getting affected generally, okay, suppose over here this boy and this boy is also getting affected, okay, then you call this, probably there are chances for it to be sex linked disorder as well. Sex linked recessive trait. As of now, it is autosomal, and as you can see that the male and the female is somehow equally being affected, which is why we can categorize it under autosomal recessive trait. You will get a hold of it. Okay. Now over here, see the difference. The parents, one parent is getting affected. One parent is clearly getting affected. And the child, again, the children, they have it. Again, when these parents, they come together, the affected child, again you have a child with affected disease out there. So that means probably it might be a dominant trait, and dominant traits, they go on every generation. Since only one chromosome is enough, if in the one chromosome you have the defect, my dear children, since it is dominant, the other one cannot overshadow the effect, which is why you will have that disease. So it is why we say that it is not going to skip the generation. So in every generation you will be able to spot this. So this would be for dominant traits. And again, how is it autosomal? How do you recognize it? Again, the male and the females almost equally getting affected. So females are getting affected more, that's not a concern. When males get affected more, that's when we are concerned about sex linked disorders out there. Okay. So yeah, so this is autosomal dominant trait myotonic dystrophy. So about which there is no explanation, but it is just given in your NCERT. So please remember it. Autosomal dominant trait myotonic dystrophy, autosomal recessive trait sickle cell anemia. Okay. In recessive trait, it will skip generation. In dominant trait, it won't skip generations out there. That's all. Perfect. Okay. Now one question related to it, and then we'll go ahead with chromosomal disorder, which is incorrect about color blindness. This is due to defect in either red or, you have to find the incorrect one, okay. This is due to defect in either a red or green cone of eye, resulting in failure to discriminate between red and green color. It is correct, right? A daughter will not normally be colorblind unless her mother is a carrier and her father is colorblind. For daughter to be colorblind, one X chromosome from the father, so father is supposed colorblind, and the mother is at least a carrier, then you can get a defective X chromosome. When both of them come together, yes, daughter can be colorblind. So that is true. Will not be normally colorblind until and unless this thing happens. So that is correct. If female has XCX, that is she is a carrier, then it is called carrier. But when male has XCY, then it will be colorblind. That is also correct. Right? And the next one, the son of a woman who carries the gene has 25% chance of being colorblind. The son of a woman who carries the gene, okay, the woman carries the gene and now she has a son. So there is 25% chances of him to be colorblind. Is that true? No, it's 50%. Because if she is a carrier, my dear children, X is there. Either this X or this X can go. Okay, in either way, 50% of the chance is there, not 25%, and this is where we get things wrong. So this is the thing that you have to spot. Okay, cool. These type of questions would be coming up. Okay, but don't be worried about anything. You're getting it sorted. Now, which of the following genotype will show the diseased condition in sickle cell anemia? Which one will be disease condition? HbA, that means it's normal. HbS, carrier. HbS, disease condition. So this is the one. Both B and C, no. So this is the one that you have to keep in mind. Cool? Cool. Now, next one. Okay, next we do not have a question. We are going with chromosomal disorders. So until now, my dear children, do you think everything is clear about Mendelian disorders? Is it okay? I'll tell you one thing, it takes time, so please relax and try to listen to me once more if at all you want, or probably once after listening to me, you can read the NCERT as well. Things would make more sense then. Okay, but don't lose your hope. If at all it's clear, put it down in the comment section. Okay, and if at all it's not clear, then also you can ask me. Now, Chromosomal disorders. I told you it's because probably the structure of the chromosome is not proper, or probably the number of chromosome is getting altered. Now it can happen because of two reasons. One is aneuploidy and the other one is polyploidy. Why do you think the chromosome number is changed or things like that is happening? Because failure of segregation of chromatids during cell division cycle results in the gain or loss of a chromosome. So usually what happens is that the chromatids which are there, okay, so it's like this, they both get separated. They both get separated. But suppose it is not happening. There is no separation happening. Both of them are going together. In that condition, what will happen, my dear children? You get an extra chromosome as well. Okay, at the end of the thing, you get an extra chromosome, or probably, suppose they are not going to a particular, suppose both of them were there and they went to one gamete. Both of them went together to one particular gamete. The next gamete, do you have anything to go? No. So, failure or segregation of chromatids during cell division cycle results in the gain or loss of what? A chromosome, and that is what you mean by aneuploidy. What do you mean by polyploidy? Polyploidy means cell is there, okay, nuclear division happened, the chromosome, everything, they divided, everything happened. Now the cell which has double amount of chromosome, they have to divide and to form two cells. From one big cell, everything got duplicated. Now it's time for the cell to divide itself into two, but that is not happening. Cytokinesis is not happening after the telophase stage. You have studied telophase, that is the last stage of mitosis, my dear children, okay, or of any cycle, mitosis or meiosis, okay. So in this particular stage, it is not happening. Then, my dear children, what will happen? There will be an increase in the whole set of chromosomes. There will be double amount of chromosomes in one cell itself. And it's not usually seen in us. It is often seen in what? Plants. Okay, polyploidy is a condition that you usually see in the case of plants. Aneuploidy is something that you see over here in us. When does aneuploidy happen? When there is failure of separation of chromatids during cell division. It can lead to either gain or loss of a chromosome. When it is leading to gain of a chromosome, it is known as trisomy. It is known as trisomy. So, trisomy means instead of having two chromosomes, now you have one more chromosome as an extra thing, because probably both of them came together. We get it? So, when an additional copy of chromosome may be included in an individual, this is known as trisomy. Monosomy is when individual may lack one of any pair of the chromosomes. Okay, since two of them came over here, probably there might be a gamete which didn't have any chromosomes at all. So, that is known as monosomy. So, trisomy is different, monosomy is different. Cool? So, that is how chromosomal disorders are being caused, and now you are going to see examples of them. So, there are three very, very important examples. One is Down syndrome, the second one is Turner syndrome, and the third one is Klinefelter syndrome. Starting with Down syndrome. What happens is that, I told you we have 23 pair of chromosomes, right? Now, the 21st chromosome, I get one from the father, one from the mother. But suppose, here I am getting two of them from one parent. So, in total, how many chromosomes are there? 3. In total, how many chromosomes are there? 3. So, the 21st one, I have 3 of them. So, extra chromosome is there. So, instead of having, we usually have 23 pair. 23 pair means a pair has 2 chromosomes. 23 into 2 is what? 46 chromosomes. We have usually 46 chromosomes, right? But over here, what is happening? Over here, what is happening? We have one extra chromosome as well. So, we put this condition as 47A and XX or XY. XX or XY. So, rather than putting it like this, you can simply put, instead of putting 47A and things like that. Understand this thing. Out of 46 chromosome, 44 of them will be autosomes, right? And 2 of them will be what? The sex chromosome. It can be XX or it can be XY. Right. So, you can write, since one extra chromosome is there, so you can write 45A plus XX or XY. We can write like that. Okay, instead of writing this, you can write like that. I'll write it and give it to you. So, we usually have 46 chromosomes. Out of that, 44 are autosomes. This is a general trend. 44 are autosomes and the rest of them are XX or XY. Now, here I have an extra autosome. So, it will be 45 autosome plus XX or XY condition. If it is XX, then it's a Down syndrome patient who is a female. If it is XY, there is a Down syndrome patient who is a male. This is an example of what? Trisomy. Trisomy, why is there an addition of chromosome up there? What are the features? You need to remember the features. Short stature would be there, round head will be there, furrowed tongue. Tongue will be like this. There will be a furrow over here in the tongue, usually. Not exactly like this, but yeah. There will be broad palm crease, mental retardation will be there, and delayed development will also be there. Okay, so there's a picture given in your NCERT. Please take a look at it. I forgot to put it over here, but you please take a look at it. It's very, very important. Okay, congenital heart disease disorder will also be there in them. So that has also been mentioned. So short stature, round head, furrowed tongue, broad palm crease, mental retardation, delayed development, and also the congenital heart disorder is also there. 45 autosomes and XX or XY condition would be there. This is an example of trisomy, and which chromosome is there? 21st chromosome. Trisomy of 21st chromosome. Now, the next syndrome is Turner syndrome. What happens in Turner syndrome? Again, it's easy. Listen, usually, okay, I have, I think, swapped the things. So this is for Turner's and this is for Klinefelter's. So I'll take Turner's one. So over here, it should have been XX condition. X from one parent, X from another parent. But you are not getting this chromosome from another parent, which is why it is now XO condition. It is now XO condition. How many chromosomes are there here, my dear children? How many chromosomes are there in this condition when you lose one chromosome? So out of 46 chromosomes, one chromosome is lost, you just have 45 chromosomes. Agreed? You just have 45 chromosomes and it will be XO condition. It will be what? XO condition. So you can just write, instead of writing like this 45 XO, you can also write 44 autosomes plus XO condition. Okay? Only one X chromosome is there is no other chromosome. So 45 plus 1, 44 plus 1, 45. Or you can also write like this, where you're saying 45 XO. But don't get confused. Don't think that autosome is extra. Autosome is not extra over here. One chromosome is missing over here, which is why what will happen? Sterility will be there. That means they won't be able to produce fertile offsprings. Okay, sterility is there. Why? Because the ovaries are not formed properly. If it was XX, then the sex chromosome there, they will be giving the sexual characteristics. Over here, since there are no two sex chromosomes out there, it will be rudimentary ovaries, my dear. Ovaries are not developed properly. This is also very much important. Okay, why they are sterile? Why Turner syndrome, human beings are sterile? Because they have rudimentary ovaries and they lack several secondary sexual traits and have short stature as well. Those organisms, so those human beings who have Turner syndrome have a condition like this. Now, next thing. So this is an example of trisomy or monosomy. This is an example of monosomy where you are losing a chromosome. Remember, Turner syndrome is an example of monosomy. Now you have one more thing, that is Klinefelter syndrome. Where what is happening? So here, my dear children, it should be XY. X from 1, Y from 1. But from the father you are getting both the chromosomes together, or probably from the mother, either way. So it is XY, and extra X chromosome is coming over here. So here, my dear children, 44 autosomes are definitely there, but XXY condition is also there. That means 44, then 45, 46, 47 chromosomes are there. So you can also write it like this, or you can write it like this, 47 XXY. There you can write 45 XO, here you can write 47 XXY. So my dear children, what will happen? It had to be a male, but now with an extra X chromosome, what will happen? Masculine appearance will be there, but with gynecomastia. That means there will be slight breast development as well. They will also be sterile because it's not proper. Something is dominating them, right? And incomplete secondary sexual development is also being there. Incomplete secondary sexual development is also being there. So that is about the Klinefelter syndrome. I'll revise it once. Listen to me carefully. Down syndrome, you are having addition of one extra 21st chromosome out there, which is why you have short stature, round head, furrowed tongue, broad brown case, mental retardation, and also delayed development along with congenital heart disease. Okay, now going ahead with this one. Also over here the condition is what? 45 autosome plus XX or XY. Now we go ahead with Turner syndrome, where you're losing one X chromosome. Basically there is one missing X chromosome, and hence rudimentary ovary will be there, lack of secondary sexual characteristic will be there, short stature would be there. So condition is 44 autosomes plus XO condition, or 45 XO condition. You can write it like that also. Klinefelter's, where you have one extra X chromosome. So it had to be a male, but now that male masculine appearance is there, but along with gynecomastia, he also would be what? Sterile. There will be incomplete secondary sexual characteristics as well. Okay, this is 47 XXY, or you can write 44 autosomes because autosome number are not been altered, and then XXY as an extra thing as well. So this is about the chromosomal disorders. So aneuploidy is where the failure of segregation is happening of chromatids during cell division. When cytokinesis itself is not happening, my dear children, that means altogether you have duplication, okay, of a whole set of chromosomes. That is totally different. That's all. And that is often seen in what? Plants. That is one very important MCQ question. Trisomy, monosomy I explained, all this thing I explained. With that, we complete what? Chromosomal disorders as well. I just kept this page in general. So now, I can club all of the diseases and put it over here. Mendelian disorders determined by alteration or mutation in the single gene or the genes which are there. Here, it is because of alteration in the structure of number of chromosomes. Color blindness, hemophilia. I'll just sum it up. Color blindness, hemophilia, both of them are what? X-linked recessive disorder. Okay. Sickle cell anemia, phenylketonuria, thalassemia, they are all what? Autosome linked recessive disorder. Recessive disorder. And then you have cystic fibrosis and also myotonic dystrophy. About myotonic dystrophy, they have given the pedigree chart. With that, we understand that it is autosomal, definitely, and it is what? Dominant disorder. In dominant disorder, do we see that in every generation? Yes. The character, you see that in every generation. In chromosomal disorders, you have three of them: Down syndrome, Turner syndrome, and also Klinefelter syndrome, my dear children. Okay? Works? Now, just give me a second. Over here. Okay, then that's all, right? Okay, one more disease is given, cystic fibrosis, my dear children. This is also coming under autosomal recessive disorder, but it is not explained in the NCERT. So one is dominant, and that is being mentioned over here. So that's all. With that, let's come to this question. Find out the correct statement, and now I am pretty sure you will be able to find it. Hemophilia is a sex-linked recessive character, is it? Yes, ma'am. Down syndrome is due to aneuploidy. So failure of segregation of chromosome. Yes, it is a trisomy condition. Phenylketonuria is an autosomal dominant gene disorder. Is it autosomal dominant gene disorder? No. It is what? Sorry. Just a second. I would have to open it again. Come on. No. Just give me a second, children. Yes. Where is it? Yeah. So over here, it is a autosomal dominant disorder. The answer is no. It is what? A recessive disorder. It is not a dominant disorder. So this is wrong statement. Now phenylketonuria is an autosomal recessive gene disorder. Correct. Sickle cell anemia is an X-linked recessive gene disorder. No, it is autosomal disorder, not X-linked. So what do you have as correct statement? A, B and D. A, B and D. Cool. And now I have a homework for you guys. What is the genetic disorder in which what happened? An individual has an overall masculine development, gynecomastia, and is sterile. Try that for yourself. Okay, that's your homework question. So today I have not given much homework question. Maybe because I thought that it might be too much for you today if I give you homework as well. Now, Sagar had a doubt about linkage and recombination. So those of you who want to live the class, you can go. I just want to explain it to him. So Sagar, if you are listening, listen to me. Linkage and recombination, I will give you an example and I will teach you. Okay, I mean I will give you whatever. Okay, pictorial representation. So over here, I say that there is A gene, B gene. Similarly, A gene, or basically I'll write capital A and capital B over here. Okay, these two genes that you see over here, they are very closely linked to each other. They are what? Tightly linked. They are very close to each other. Now, if I want to swap their positions, suppose this was like this and this is just normal. If I want to swap the position, what will happen, my dear children? A and B will still be together, capital A and capital B will still together, and small a and small b will also be together. Why? Because they are very close to each other. So whenever transfer is happening, they are getting transferred together itself. So if we talk about the new combination that is being formed, so the answer would be new combination is definitely being formed. Why? Because here I am changing the two parts. So this was with this now, but this is now with capital A and capital B. So recombination is definitely happening, but the amount of recombination, isn't it less? Why? Because these genes were always together. Now in the same condition, if I show you something else, okay, there is small a, small b gene, there is capital A and capital B gene, okay, and over here there is some change, and over here also it's like this, okay. Now they are far away from each other, so I'll say that they are loosely linked genes. That means they are very far away from each other. Now when recombination swapping happens, crossing over happens. Crossing over is ultimately very important procedure that happens during cell division, okay. So when the swap happens, my dear children, suppose this remains the same. Over here it counts this side. Now capital B is over here. And if this goes that side, then small b is over here. And what happens? Capital A will be over here. Don't you think it's an ultimately complete change? This and this came together. This and this came together. So here, if you see the recombination that has taken place, my dear children, here, if you see the recombination that has taken place, it will be way too more. There is change over here also, there is change over here also, and very big changes that you can see. That means what? Loosely linked genes shows higher recombination. Tightly linked genes which have shorter distance between them, they show lesser recombination. Okay, that's the whole thing, nothing else. So if at all in the question it is said that 37.2% recombination is there, just understand it is too much recombination, and just understand that they are far away from each other, they are loosely linked. If it is said that something is showing 1.3% recombination, just understand the genes are very close to each other, they are tightly linked to each other. That's all. Okay, that's all, my dear children. With that I complete this. I know I took a lot of your time, but this is a very, very important topic, so I wanted to teach you. If you still have some doubts, put it down in the comment section. I am here to help you with that. That's all. And do not forget to subscribe to the channel also if at all you have not subscribed yet. Wishing you all the best. Bye-bye. Take care.